Homeostasis is the first concept you will learn in medical physiology. It's more than a concept, it's going to be the underlying theme of your entire physiology course. Today we are going to discuss this homeostasis in a conventional way, that is how it was originally proposed by Claude Bernard, that is stability of internal environment. So what he suggested as internal environment, let's start with a single cell organism like bacteria. Bacteria can survive in extreme temperatures, like different types of bacteria adapt to different environmental conditions. Like some bacteria live under boiling temperatures, and some bacteria lives in freezing conditions like less than 0 degrees centigrade. Different species of bacteria has adapted to survive under different environmental conditions like temperature, oxygen, pH, and so many other things. Though they have ability to adapt to a different condition over a period of time, a given bacteria, if for example these bacteria, cryophilic bacteria we call them, which lives under sub-zero temperature, if we put them on a higher temperature, they may not thrive. A single bacteria has evolved to survive only under a particular environmental condition. So if we change it, it is going to die. But when it comes to multicellular organisms, they create a micro environment surrounding their cells. We call it as an internal environment. It actually their body. So this internal environment here, as these are the group of cells, and this is a body of the organism, and you can see this is an external to the body. So there is a controlled environment surrounding their cells, which is called as the internal environment, and the actual surrounding of the entire organism is called as a external environment. In the multicellular organisms, try to control the internal environments variables like oxygen level, temperature, pH, and so many other things. This is called as the stability of life conditions in the internal environment. If you take a human body, it has trillions of cells, like the cells inside of different organs are going to perform different functions. Like an RBC is going to transport oxygen, a nerve cell is going to transfer information to other cells, and a muscle cell is going to produce force. So there are many cells which will perform different functions, but collectively they are all trying to maintain the conditions in the internal environment as a stable constant one. And it's surrounded by the protective cells, and there are exchange cells which will transfer or exchange energy and biomolecules with the environment. This is the conventional way of looking at it, that is the internal environment means extracellular fluid and where the cells and other things survive, and the homeostasis is maintaining the conditions of the extracellular fluid, that is internal environment. So this is the conventional way of looking at it, though today we understand that even single cellular organisms maintain their own internal environment that is within the cells. It is also considered as a homeostasis. So now we understand that entire body, not just the extracellular fluid, is considered as an internal environment. The cells produce it, and they all take the benefit out of it also. They survive within this constant life conditions. This was originally proposed by Claude Bernard. He is doing an experiment over here. He is a big fan of experimental science, and he is also called as a father of modern physiology. Let's see what he has said about this concept. Maintenance of stable milieu interieur. Milieu interieur is French, and in English it is internal environment. He has said all the vital mechanisms, however varied they may be. Here we are referring to all vital mechanisms like respiration, excretion, locomotion, and all the other functions. However varied they may be, have one common goal, that is to maintain the uniformity of the conditions of the internal environment. The life conditions we are referring here as this oxygen level, pH, temperature, glucose, and all these things. So he said the stability of this internal environment is the condition for free and independent life. Though Claude Bernard gave the concept, the word homeostasis was coined by Walter Cannon. He further expanded the concept and popularized it in his book Wisdom of Life, and he also coined the term flight or fight response also. Let's see what he has said in his book. Some of the important ideas were: constancy in an open system requires mechanisms that act to maintain the system. Open system here referred to the fact that human body continuously exchanges like substances and energy with the external environment. If something is called as a closed system, then there is nothing is exchanged with the environment. So human body is an open system because continuously substances are exchanged with the environment. So such system is called as a steady state. Like a system has a stable internal environment, but there is continuous exchange with the environment happening all the time. A stable state happens even in another condition called as an equilibrium, where the internal environment of a system is constant because there is no net exchange happening between the system and its environment. Equilibrium is not compatible with life because to sustain life processes, energy is required, which must be gained from the environment, and other things like waste products are to be lost to the environment. So the condition maintained within the body, that is a constant internal environment, despite the fact that there is a continuous exchange happens with the environment, this is called as a steady state. And steady state conditions require that tendency towards change are automatically met with factors that resist the change. Steady state indicates that a variable in a system is kept constant though it is continuously being added or removed from the system. Maintenance of steady state requires energy and a control system which matches the input and output of the variable. For example, the temperature of a human body is maintained at 37 degrees by balancing continuous heat production by the human body and heat loss to the surroundings. Any maintained variable, including the temperature, cannot be exactly constant, but it is maintained within the narrow range, so-called a relatively constant value. So that's why it is used in the definition of homeostasis. Homeostasis is the maintenance of steady state, that is a relatively constant internal physical and chemical conditions in the body by coordinated physiological control system. So he also adds homeostasis does not occur by chance but is a result of organized self-government. And yes, this is true, and that is what is the function of a homeostatic control system. Let's see what it is. Homeostasis involve dynamic mechanism that detect and respond to deviations in physiological variables from their set point values by initiating an effector response that restore the variables to the original optimal physiological range. So these are the key words in this definition. We are going to understand this by using an example of air conditioner. The air conditioner in your room is maintaining a variable, that is room temperature, and let's say the room temperature is 30 degrees and you set it up at 28 degrees through your remote. So the value you are setting is the set point and the actual level of the variable is 30. Once this is set, the exact value is detected by things called a sensor, which is going to detect what is the actual value and it is going to compare with the set point, and if there is a difference then it is going to generate a response, that is the AC is going to generate a response of sending cool air into the room, so thereby eventually it restores the room temperature. And when it restores, it's not going to be exactly maintained at 28 degrees centigrade. There may be a small range. For example, if it goes below 27, it may switch off, and then it may go above 29, then it may start working. So there is a range in which the room temperature is maintained. Though we are giving a set point of 28, there is an optimal range in which the room temperature is maintained. And this is the explanation for all the key points in a homeostatic control system. Let's discuss the components of homeostatic control system. As we discussed above, there is a sensor which is going to detect changes in the variable, and it is going to send this information to a feedback controller, which is a part of the central comparator, and it compares set point value with the sensor detected value, and if there is a change then it is going to send the information to the effector, which is going to influence the regulator variable again, which is this is called as a feedback loop. And there are two types. One is a positive and a negative feedback loop. Let's see each of the things with an example. Let's say there is a regulator variable at a particular level. There is a disturbance which causes the regulated variable to increase, and the sensor detects the change, sends to the feedback controller, and it detects the difference, and now it initiates an effector response to put down this regulator variable so that it comes back to its normal level again. Let's see this negative feedback with an example of plasma glucose level maintenance. Here the regulated variable is plasma glucose. Let's consider a disturbance, that is consumption of food, which cause the glucose levels to increase. And this is sensed by the sensor, which is located in the beta cells within the pancreas, and it's going to sense the difference between the set point and the set point value and the actual value, and if it is higher, the actual value is higher, it is going to release insulin. So this is the pancreatic beta cell, which is going to function as the sensor and the feedback controller, and the effector is the insulin. The insulin is going to act on various body tissues and going to bring the glucose level down. So here original change is increased, but actual response of this control system is to minimize the change. So this is the basic concept of a negative feedback system. In a negative feedback system, the stability is maintained. A positive feedback, there is a regulator variable, and if a disturbance causes it to increase, which is detected by a sensor and feedback controller, and ultimately the effector is going to again cause more and more increase. It is going to do in a same direction. Whatever the original change direction, the effector is also going to augment this change. Let's see this with an example. Opening of voltage gated sodium channel, NAV stands for voltage gated sodium channel, in action potential. Here the regulated variable is membrane potential. Membrane potential is going to be maintained at minus 70 millivolt. But when there is a small depolarization disturbance, this is going to be sensed by sensor within the voltage gated sodium channels. We call it as a voltage sensor, and this is going to communicate with the other components of this voltage gated sodium channel, and they are going to open this. When the NAV opens, it's going to cause more and more depolarization. So if the original change is from minus 70 to minus 60, then opening of more and more sodium channels is going to depolarize it to, let's say, 0 or even plus 10 millivolts. So this is what happens during an action potential. This is an example of positive feedback. So positive feedback is essential for promoting a change in the system. They are not essential for maintaining the stability as such of the variable, but they promote change in a condition. But this is essential in a large picture. This is essential for the homeostasis or the survival of the organism. Even the positive feedbacks are essential, though sometimes the positive feedbacks can work in a vicious cycle and may produce deleterious effect. Let's see one other mechanism, that is feed forward. Past two mechanisms are feedback loops. Now this is a feed forward mechanisms, and this is glucagon like polypeptide 1 induced insulin release. Here the regulated variable is again glucose, and this is first I'm going to draw the feedback loop. When there is a disturbance, like when is food consumption, and glucose levels are sensed by the insulin and ultimately the beta cells, and ultimately it is going to release the insulin and going to decrease the glucose level. This is a feedback loop. But sometimes even before the change happens, the disturbances which are causing the change in the regulated variable are sensed by other controllers, like a feed forward controller, which is going to influence this regulated variable through either a feedback controller or a effector. That here directly insulin is released by the presence of food in the intestine. Some cells sense the presence of food in the intestine and releases GLP-1, which is going to stimulate the insulin release. Though the regulated variable is glucose, which has not changed yet, Even before the change happens, the glucose levels are going to be regulated by the insulin levels. So this is called as a feed forward mechanisms. Here, though the feedback mechanisms, both positive and negative, operates only when the regulated variable changes. But even before the regulated variable change, the disturbance itself is sensed by a feed forward controller and it proactively maintains the system. Sometimes this is referred as anticipatory control system. Okay, so thanks for watching. Our next lecture is on negative feedback gain. Stay tuned.